Thanks for stopping by to learn about folding at home. We're a community of scientists and citizen scientists working together to drive biomedical research and combat global health threats from COVID-19 to Alzheimer's disease, cancer, and beyond. One of the unifying themes behind our work is an obsession with understanding the moving parts of proteins, which are the molecular machines responsible for many of the active processes that we associate with life, ranging from muscle contraction to sensing light in the eye and many other biological processes. Unfortunately, it's impossible to watch the moving parts of these molecular machines with existing experimental techniques. And so to fill this gap, we're trying to simulate protein dynamics on computers. One of the challenges is that these calculations are extremely computationally demanding though. So we simply can't do what we need to do on a single desktop computer or even on national supercomputing resources. And so as an alternative, what we've done is gone out on the internet and invited anyone with a computer and an internet connection to become a citizen scientist and volunteer to run computer simulations on their personal computers and send the data back to our scientific team. And this has been immensely successful over the past 20 years. In fact, during our work on the SARS-CoV-2 virus, over 1 million devices were participating in folding at home, each represented by one of these yellow dots on the map to my side. And this provided an immense aggregate compute resource, the first to reach the exascale, providing over 5 times the performance of the world's fastest supercomputer at the time. And with this, we were able to do a lot of really great science. Just as one concrete example, this is a representation of the spike complex that's protruding from the surface of the SARS-CoV-2 virus in the images that I'm sure you've all seen. So in the upper left is where this complex of proteins is tethered to the surface of the virus. And in the lower right is where this complex has to open up in order to bind to a protein on a human cell and initiate infection. One of the challenges in this specific case is that experiments had observed this closed up structure, but we really didn't know what the open structure that initiates infection looks like. But here's what we've learned in a qualitative sense from our simulations. And so what you can see is this dramatic opening up of this complex that exposes the interface that has to bind to the ACE2 receptor on human cells to initiate infection. And the structural information from our simulations is now helping to inform the development of future vaccines and potentially of therapeutics for combating the SARS-CoV-2 virus that is responsible for the COVID-19 pandemic. We've worked on lots of other proteins as well, and going beyond the science, our community has really done a lot to build on the platform that we've created, including creating a Discord server where they can discuss the science and the best hardware to contribute to folding at home, cryptocurrencies that reward people for participating in the project, and many other grassroots efforts. So I invite you to learn more from the poster here and also from our website at foldingathome.org or our social media channels like our Twitter account at Folding at Home. And you're welcome to reach out to us through any of the means on our contact page online. Welcome, and we hope that you'll soon be a participant in our massive distributed computing project.